Hello everyone, my name is Yulong, a PhD candidate at University of Ottawa. I study in molecular evolution. One research area I focus on is how translation efficiency affects bacterial growth and replication. Understanding factors that optimize translation not only advance our knowledge of molecular biology, but also improve the efficacy of recombinant DNA in transgenic biosynthesis. To improve translation efficiency, protein coding genes co-evolve with the translation machinery. Today, I talk about the Shine-Dalgarno mechanism that affects translation initiation process in two specific organisms, Cyanobacteria and Chloroplast. And the title of my presentation is An Improved Model for Shine-Dalgarno Mechanism Reviews Evolutionary Differences in Cyanobacteria and Chloroplast Translation Initiation. A bit of background on the Shine-Dalgarno sequence. The motif is prominently found in prokaryote species, hereafter referred to as the SD sequence. How it works is illustrated in this figure here. In the 5' untranslated region upstream of the start codon of bacterial genes, we often find a purine-rich sequence motif. The location of this motif can vary between genes, illustrated as SD1 and SD2, and their distance from the start codon may differ in different genes. The SD motif pairs with the anti-SD motif that is located at the 3' end of the 16S ribosomal RNA. This pairing does two things. First, it facilitates the recruitment of the ribosome to the mRNA. Second, the specific pairing between SD and anti-SD sequence allows for the proper juxtapositioning between the start codon and the active A site on the ribosome. As a result, genes that have an SD sequence have better translation initiation efficiency than genes that don't have the SD sequence. A good SD sequence that facilitates translation initiation therefore needs to meet two criteria. First, it has to have good pairing potential with the anti-SD sequence. Second, it is located at the optimal distance to allow proper positioning between the start codon and the ribosomal A site. In my presentation, I will focus on the second criteria, which also happens to be the more important feature. In our previous studies, people have measured this distance as how close to or how far away the SD motif itself is to the start codon. However, there are problems with this conventional measurement. Here we have 6 E. coli genes, and using this measurement, the location of the SD motif in bold will all differ. However, if we were to instead measure the pairing location from the 16S ribosomal RNA end, we see that the distance at the bottom is well conserved across all 6 genes. It also makes more sense to measure the distance at the bottom here because what matters in this interaction is whether the pairing will allow proper positioning between the start codon and the ribosomal active site. It doesn't matter where the SD sequence itself is located, so long as the specific pairing places the ribosome near the same locations. We coined this new distance measurement as D to start, and when we took the entire E. coli genome, determined all SD-containing genes, then measured the distribution of these two start distances, we see that this distance is evolutionarily conserved across all genes. Here is another graph that shows that D to start constraint is indeed important. When translation initiation is efficient, we can expect faster ribosome loading onto the mRNA. Genes with efficient translation initiation should have increased ribosome abundance at the initiation region, in comparison to genes with inefficient translation initiation. We made this comparison by mapping ribosome protected reads on all genes having an SD sequence in red and all genes that lack an SD sequence in blue, in E. coli and Bacillus subtilis. Here the start codons are located at site 60, and as we can see, the increase in ribosome abundance in SD-containing genes precisely corresponds to the location of the optimal D to start location in yellow. So this shows that our improved SD location measurement is indeed important for translation initiation. Now comes the challenge in this approach. We often do not know the precise 3' end of the 16S ribosomal RNA. This has to do with the fact that we do not fully understand how the 16S RNA post-transcriptional modification process works. For us, the problem is if we do not know the extent of this tail sequence, then we cannot detect the complete pool of SD sequences. And when a partial anti-SD 3' tail sequence we cannot determine the optimal D2 star distance. Without these information, we cannot determine whether the SD mechanism is important for a given species. To resolve this problem, we use transcript profiling using RNA sequencing data. Without going into too much detail, the logic here is straightforward. We align RNA sequencing fragments to genomic sequences, and by determining the distribution of these rates onto the 16S ribosomal RNA sequence, we can find where exactly the 3' end is. So, using this approach, here we show what the 16S ribosomal RNA strip-on tail sequence look like in Bacillus subtilis. As you can see in panel B, there is pretty convincing evidence that the red bar here represents the mature strip-on tail sequence in this species. And we did the same for about 20 more bacterial species, and in red are species where we found errors in NCBI annotations. So in this, we propose an improved approach to determine what is an optimal SD sequence in any given species. But all models are useless unless they have practical uses. Here we applied our model on a special case, the translation initiation process in cyanobacteria and chloroplast. Previous research have shown that the SD sequence is less prominently found in chloroplast. Its location was not well conserved. Mutating the SD sequence or deleting the 5' untranslated region entirely did not strongly affect chloroplast gene expression. These led to further findings that chloroplasts have unique ribosomal proteins, and its translation initiation may require a different transacting factor. This has also led to the debate on the centrality of SD sequences in cyanobacteria, because chloroplast presumably originated from cyanobacteria. So the question is, was the role of SD mechanism weakened in cyanobacteria prior to chloroplast endosybiosis, or did chloroplast lost this SD mechanism when it evolved away from its bacterial ancestor during endosymbiosis? Contrasting SD usage between these two species therefore have a distinct importance to understand the evolution of prokaryotic translation machinery. We predicted that if genes in both species show weak SD usage, then SD function may have been reduced in cyanobacteria prior to chloroplast endosymbiosis. However, if SD usage is important in cyanobacteria but not in chloroplast, then SD function may have been reduced in chloroplast after it has differentially evolved under host-symbiont coevolution. Here are some key findings in our research. In panels A and B, we see that D2 star constraints are highly similar in two cyanobacterial species, Synechocystis species in blue and Microcystis aeruginosa in orange. However, their D2 star constraints are dissimilar from two chloroplast species shaded in gray in panels A and B. Panel C and D shows that in contrast, cyanophages SSM6A and SSM6B well mimic host cyanobacteria in D2 star constraint. The importance of this finding here is that phages utilize host translation machinery to replicate. Therefore, it is evolutionarily beneficial for phage genes to adapt host gene features that increase translation efficiency. So, because the SD location in cyanobacteria is well mimicked by cyanophages, SD sequence must be important in cyanobacteria. This figure here shows us that, indeed, SD-facilitated genes have higher protein expression levels than SD-lacking genes in cyanobacteria in panel A, but not in chloroplast in panel B. In conclusion, cyanobacteria and chloroplasts differ in the utilization of SD sequences. In cyanobacteria, SD usage is still important. These sequences exhibit similar preferences for pairing and location as in E. coli. Cyanobacteria SD usages are well mimicked by cyanophages, and SD usage increase gene expression. However, these characteristics were not observed in chloroplast. Using our improved SD model, we were able to resolve the long-time debate over the importance and difference of SD sequence in cyanobacteria and chloroplast. And these results support the hypothesis that SD function has been reduced in chloroplasts after having differentially evolved under host-symbiont coevolution. And this research may serve as an anchor for continued study in chloroplast translation initiation in order to understand how translation machinery is subject to change in plastids that have evolved away from its bacterial ancestor. And that is the end of my presentation. Thank you everyone for listening.